Hello everyone, welcome back to my YouTube channel Biology Colony. In this lecture, we are going to discuss the previous year questions of GATE Zoology 2022. Okay, so the first question is, which one of the following organic compounds is composed of only a nitrogen containing base, a single 5 carbon sugar and a triphosphate? The answer will be nucleotide. As we know that nucleotides have three components, nitrogenous base, pentose and a phosphate. As you can see in this picture, it is a phosphate group, a sugar and a nitrogenous base. Then nucleoside is the one without the phosphate group. So nucleotide minus phosphate makes up the nucleoside. Nucleic acids are long chain polymeric molecules. It is known and the monomers are known as the nucleotides, and hence sometimes nucleic acids are referred to as polynucleotides. Then we have the nitrogenous bases. They are of two types, pyrimidines and purines. And both DNA and RNA contain two major purine bases, that is adenine and guanine, while RNA contains cytosine and uracil and DNA contains cytosine and thymine. The next question is, which one of the following animals develop adaptive predator avoidance morphology because of the presence of high predator number in its habitat? So the answer will be Daphnia species. It has been observed in this species that they migrate or they develop neck spines or change their body size. These are a few avoidance morphologies or adaptations. They were shown by the Daphnia species due to high predator number in its habitat. So the next question is, to which class of Drosophila developmental genes does Fushi-Tarazu belong? Gap genes, segment polarity genes, pair rule genes, or maternal effect genes? Well, the answer is pair rule genes. This is a very important concept of Drosophila development biology. This is a table taken from Developmental Biology by Gilbert. It is a very important table. You have to remember these. As you can see here, Fushi-Tarazu is a pair rule gene, secondary pair rule gene. This is a very important flowchart. Next we have the action of which class of enzyme inhibitors can be reversed by adding an excess of substrate. Uncompetitive inhibitors, competitive, non-specific or allosteric? Well, the answer is competitive inhibitors. Enzyme inhibitors are molecular agents that interfere with catalysis, slowing or halting enzymatic reactions. These are of two types, reversible or irreversible. As the name suggests, reversible means the inhibition can be reversed, while in irreversible it cannot be reversed. So competitive inhibitor competes with a substrate for the active site of an enzyme. You can see in this diagram. This is the active site of an enzyme, and as you can see, substrate is bound with the enzyme here, and then enzyme is bound with the inhibitor at the same site. This is a competitive inhibition. So while the inhibitor I occupies the active site, it prevents binding of the substrate to the enzyme. तो जब ये substrate से bind नहीं कर रहा है, I से bind कर रहा है, तो substrate इसमें bind नहीं कर सकता है. So many competitive inhibitors are compounds that resemble the substrate. As इनकी same site है, तो इनका structure भी थोड़ा similar सा होएगा. So because the inhibitor binds reversibly to the enzyme, the competition can be biased to favor the substrate simply by adding more substrate. जैसे ही हम ज़्यादा substrate डालेंगे तो inhibitor का number क्या हो जाएगा, कम हो जाएगा. So substrate can bind to more number of enzymes. Thus the reaction may proceed or the catalysis can occur. When substrate concentration far exceeds inhibitor concentration, the probability that an inhibitor molecule will bind to the enzyme is minimized and the reaction will exhibit a normal Vmax. Then the other two are uncompetitive inhibition and mixed inhibition. These are also reversible type of inhibition. As you can see in the uncompetitive, the active site of the substrate differs from the binding site of the inhibitor. So inhibitor binds to the other site, that is the site other than the active site. Then in mixed inhibition, it also has the site other than the substrate site. So it is a mixed inhibition. Next question is, Mendel deduced the genetic principle of inheritance by experimenting on pea plants, sweet pea plants. One of the experiments involved crossing plants with two contrasting characters, that is tall and dwarf, that is recessive, and which yielded all tall plants in the first generation. F1 has all the tall plants. When the same genetic cross was independently repeated by a researcher, only short plants were obtained. Which one of the following can possibly explain the altered outcome? Now we have to deduce the answer from the options. Tall plants were heterozygous, means they were capital T and small t. If this is the case, then tall must appear, but it is not showing in the experiment done by the researcher, so this option is incorrect. An enhancer for the tall allele is present in the dwarf plant. If enhancer is present, then tall would definitely appear, but this is not the case, so it is also wrong. A suppressor for the tall allele is present in the dwarf plant. Yes, there is a suppressor present that is preventing the tall expression to appear. Then D is dwarf plants are homozygous. Okay, they can be homozygous, but it does not mean that tall will appear. Which of the following is or are responsible for reversible receptor ligand or ligand interaction? Ionic interactions, hydrogen bonding, peptide bonding, and hydrophobic interactions. This is a multiple select question, so more than one option is correct. In this case, ionic interactions, hydrogen bonding, and hydrophobic interactions are responsible for reversible receptor ligand interaction because these are the weak bonds. They can be broken up easily, while the peptide bond, it is a covalent bond, that is a strong interaction. So the answer is A, B, and D. In this question, in the human body, which of the following is or are involved in processing of a foreign antigen? Foreign antigen processing is done by B cells and macrophages, while red blood cells do not perform such function, while platelets are involved in blood clotting. So the B cell or B lymphocytes is a key player for the adaptive immune response that is responsible for humoral immunity in mammals. It acts by producing antibodies, antigen presenting cells that are APCs, supporting other mononuclear cells and contributing to inflammatory pathways directly. So B cells are involved in antibody production, which are basically binding to the antigens and are processing. Then macrophages are effector cells of the innate immune system that phagocytose bacteria and secrete both pro-inflammatory and antimicrobial mediators. In addition, macrophages play an important role in eliminating diseased and damaged cells through PCD or programmed cell death. So macrophages are also antigen processing cells. Then dendritic cells, they have the broadest range of antigen presentation and are necessary for activation of naive T cells. So what happens? These B cell, macrophages and dendritic cells, they process the antigen and then are presenting the antigen to the T cells for further processes. So the next MSQ question is, animals can be classified as specialists or generalists with respect to diet and habitat selection. Which of the following organisms belongs to the specialist category? Specialist category पूछा हुआ है. तो raccoon, these are the generalist. While panda, polar bear and koala bear, these are the specialists. So let's understand the terms generalist and specialist species. So a generalist species is able to thrive in a wide variety of environmental conditions and can make use of variety of different resources. So they have a varied diet. Example is raccoons. They live throughout the North and Central America. They are omnivores and they are able to survive in variety of environmental conditions, and thus it has enabled them to maintain large population size. While specialist species, they require very unique resources. They have very limited diet and they have narrow range of habitat. So examples are polar bear, they survive in cold climate only, while pandas, they are herbivore and they eat only bamboos. And koala, these are herbivorous marsupial mammals found in Australia and they feed only on eucalyptus leaves. So these are the example and definition of generalist and specialist. Okay, so next we have match the following. Match the drug/chemicals listed in column 1 with the developmental/physiological defects listed in column 2. First is veratrum alkaloids. This is cyclopia. It causes cyclopia. Then thalidomide. It causes phocomelia. Then we have methyl mercury. It causes Mina-Mata syndrome, while diethylstilbestrol causes obesity. So the option first is correct. So cyclopamine is a naturally occurring steroidal alkaloid, or it is a veratrum alkaloid. It is a component of corn lily. It is a teratogenic component of corn lily, a plant called Veratrum californicum, which when consumed during gestation has been demonstrated to induce birth defects, including the development of single eye, that is cyclopia. Mina-Mata disease, it is a neurological disease caused by mercury poisoning. In this case, it is caused by methyl mercury consumption. So these are the two diseases. Then this table is very important. You must learn this slide. It is taken from Gilbert, Developmental Biology. These are the drugs and chemicals or the ionizing radiations or other microorganisms or metabolic conditions that can cause teratogenic effect. Then these are the human teratogenic drug. We talked about thalidomide that causes phocomelia, that is the loss of limbs. And then anti-cancerous drugs, androgens, progesterones. We talked about stilbestrol and tetracyclines, warfarin. These are the alcohol is also one of them. So these are all the teratogenic drugs with their abnormalities. So the next question is, match the animals listed in column 1 with primary tissue or organ of residence in the host listed in column 2. So these are all the parasitic organisms given in column 1. We have to match the locations. So first is Ascaris lumbricoides. It resides in small intestine. Then we have Dracunculus medinensis. It resides in the subcutaneous tissue in human. Then Enterobius vermicularis, cecum or vermiform appendix. Then we have Wuchereria bancrofti in lymphatic vessels and lymph nodes. Wuchereria bancrofti is repeated in 2023 zoology question paper. So you must remember these parasitic organisms with their names and where they reside and their disease they cause. So option D is correct. So the next question is also match the following. Match the cell types listed in column 1 with their sources in column 2 and the primary functional roles listed in column 3. So these are the cells found in our body. Microglial cells. These are present in brain and their function is to phagocytosis. Then Leydig cells. It is a very easy one. It is present in testes और ये hormone secretion का काम करते हैं, that is testosterone secretion. Then on cells present in eyes and eyes में visual transduction में involved है. Then we have pneumocytes. Pneumo term से समझ आना चाहिए कि lungs की बात हो रही है. So और lungs से बात हो रही है तो gaseous exchange में ये help कर रहे हैं. So option D is correct. So the next question is also match the following question. So match the ecological concepts listed in column 1 with their definitions listed in column 2. So these are the ecological concepts. First is dominance hierarchies. It is a preferential access to the food and mates in a group. Preferential मतलब कि जो भी dominant होगा वो सबसे पहले food और mate के लिए आगे आएगा और उसको access मिल जाएगा. Then we have, तो ये hierarchy से होती है, ठीक है. Then territory में क्या है? Exclusion of competing individuals using agonistic behavior. First of all, what is agonistic behavior? It is by fighting. So individuals fight, they compete for a territory. Okay. Then क्या हो जाता है? Exclusion हो जाता है, मतलब कि जो competing जिससे वो compete कर रहा है उसका बाहर निकाला हो जाता है. Then altruism. What is altruism? It is giving up one's own reproductive potential to benefit another individual. So basically it is a sacrifice. Then kin selection. Selection acting on related animals which affects the fitness of an individual. It is a kin selection. So option B is correct. So the next question is, match the hormones listed in column 1 with their primary source tissues in column 2 and the primary target tissues. So we have epinephrine. Epinephrine is secreted from chromaffin tissues. These chromaffin tissues are found in adrenal, medulla, and the target organ or tissue is heart. Then we have prolactin. Prolactin is secreted from pituitary and its target tissue is pigeon's crop. So it is involved in milk production. Then we have calcitonin. It is secreted from C cells of thyroid and its target tissue is bone. Then we have thyrotropin releasing hormone. It is secreted from hypothalamus and the target tissue is pituitary. So option C is correct. So this question is an MSQ. It carries 2 marks. So 2-deoxyglucose, 2-DG, inhibits the proliferation of cells and hence finds use as an anti-cancer agent. It is also used in COVID therapy where it blocks hyperproliferation of virus-impacted cells. Mechanistically, 2-DG blocks glycolysis by inhibiting the activities of which of the following enzymes? Hexokinase, glucose 6-phosphate isomerase, glucose 6-phosphate dehydrogenase, or phosphofructokinase. So the answer is hexokinase and glucose 6-phosphate isomerase. So the reason is that acting as a D-glucose mimic means it is structurally similar to the glucose and thus it inhibits the glycolysis due to the formation and intracellular accumulation of 2-deoxy-D-glucose 6-phosphate or 2-DG6P. What happens, as you can see in this diagram, it is 2-DG mimicking itself as glucose, undergoes phosphorylation and forms 2-DG6P or 2-deoxyglucose 6-phosphate. So this glucose 6-glucose phosphate, it is accumulated in the cells and it inhibits the hexokinase and phosphoglucose isomerase or glucose 6-phosphate isomerase and induces the cell death. Thus it is used in cancer as well as COVID therapy. This is also an MSQ and it carries 2 marks. According to Ebb's equation on microscopy, the ability to resolve 2 entities inside a cell by light microscopy depends on which of the following factors? Magnification of the objective lens? No. Intensity of incident light? No. Wavelength? Yes. And numerical aperture of the objective lens? Yes. So the formula is 0.61 lambda divided by n sine theta. Here lambda is wavelength while theta, n sine theta, is the numerical aperture of the lens. You can see here this is the Ebb's equation. It is 0.612 lambda divided by n sine theta or alpha. Match the animal inactivity behaviors listed in column 1 with representative animals in column 2 and the definitions listed in column 3. So column 1 में torpor is shown by hummingbirds and it is the decreased metabolism with lowered body temperature occurring in daily activity cycles. Then we have hibernation. Hibernation is shown by ground squirrels and it is the decreased metabolism and lower body temperature for weeks or months. In winters, squirrels perform hibernation. Then we have winter sleep. Winter sleep is shown by polar bears. It is the prolonged period of inactivity without reducing the body temperature. अगर body temperature reduce हो गया तो polar bear survive नहीं कर पाएंगे. They have to maintain their warmth also. Then estivation. It is shown by the Australian burrowing frogs and it is the inactivity period which accompanies the extended periods of dryness. So option B is correct. So the next question is numerical answer type question and it carries 2 marks. If the vital capacity of an individual is 4900 ml and the tidal volume is 500 ml and the inspiratory reserve volume is 3300 ml, the ERV or expiratory reserve volume of the individual is, तो इसका फॉर्मूला जैसे कि हम जानते हैं vital capacity is equal to tidal capacity plus IRV plus ERV. So ERV can be equal to vital capacity minus tidal capacity minus IRV. So ERV is equal to 4900 minus 500 minus 3300 ml. So 4900 minus 3800 ml gives us 1100 ml as ERV. So the answer is 1100 ml. In this slide, you can see all the formulas for the lung capacities. IC, that is inspiratory capacity, functional residual capacity, vital capacity, and total lung capacity. So you should learn on all these formulas for the exam point of view. So the question is, a typical food chain involves producers, herbivores, primary carnivores, and secondary carnivores. Based on Lindeman's law of trophic efficiency, if producers have 40 kJ of energy, the energy that will be stored in secondary carnivores is what kilojoules? So what does the Lindeman's law of trophic efficiency say, or it is also known as the 10% law? It means 10% of the energy is transferred to the next trophic levels. We are aware of the trophic pyramid. Then we have producers, we have primary consumers, that are herbivores, then we have secondary consumers or primary carnivores. Then we have tertiary consumers, or then we have fourth trophic level, tertiary consumers or secondary carnivores. So what happens? The producers have 40 kilojoule of energy. So 10% of 40 joule is transferred to the primary consumer or the herbivores. Then they have 4 kilojoule of energy. Then for the primary carnivores take up 10% of energy, that is 0.4 kilojoule. Then again secondary carnivores, they have 0.04 kilojoule. So the answer is 0.04 kilojoule of energy. This slide explains the 10% law, and it states that 10% of energy in a food chain is transferred from one trophic level to another. The rest of the energy is utilized in metabolic processes and released as heat. The next question is, the average body length of Drosophila nasuta collected from Andaman and Nicobar Islands is 2 mm. From this population, a few males and a few females having a body length of 3 mm were selected and interbred. The average body length of the resultant progeny was 2.5 mm. The heritability or h-square of the body length in this population is. So we can solve this question by using this formula, which is heritability is equal to number of offspring minus mean yield of the population divided by parental yield minus the mean yield of the population. In the question, the offspring yield is given as 2.5 mm and the mean yield of the population is given as 2 mm. Then parental yield is given as 3 mm and again the mean yield of the population is 2 mm. So 0.5 divided by 1 or 0.5 is the heritability. Thank you so much for watching this video, and subscribe my YouTube channel and click on the bell icon for more updates. Thank you.